Hi, this is Miss Litton, and this is a small little group of honors biologists. I don't have a notebook presentation open. There's so many in this, so I don't know that I have individual questions that we can do clickified, but you're welcome to go back through the individual chapter reviews and use those clicker questions. That's what I also say when I'm reviewing for the AP final. There's so much information. There's 2 units. So let's go through all the main ticket, big ticket items and make sure we understand those and go over them, and we can kind of, if you're stressed on one thing, we can talk about it. So in Unit 1, there were basically 3 chapters, and they all had to do with ecology, and you had 4 big driving questions. And so if we look, there was chapter 33. If you're going to go back through and do some practice questions on that. So chapter 33 talked about ecosystems, and it also talked about how different populations interact with other populations in an ecosystem. So big things on there is you want to make sure you understand. You got this tight. Let me see. If I was going to point out some big things that one might have questions, I would say things like knowing the definition of ecology. Okay, wait, that is just not going to work. Go away, pen. Let's go smaller. So definitely make sure you understand what ecology is. And then some other topics we talked about are characteristics of populations. So on that, we talked about distribution patterns. Do you remember when we talked about the different types of patterns, what was most likely of all the distribution patterns? Remember that about clumped and random? Does this sound familiar to you? Most organisms tend to be clumped around their resources. Do you remember that? This is when we started off with the bear, and you guys were bears, remember? Way back. It's a good thing we're doing the Octodoc, because you're going to hit this chapter eventually doing something, okay? But this is when we talked about the bears and you were out competing for food. That's how we started up on it. So on distribution patterns, we talked about that most organisms tend to be clumped around their resources, not random and not uniform, if you remember that. Uniform is like equal distance between each organism, and random is it doesn't matter if you're here or there. That would mean low competition if it was in that situation. Another characteristic of populations, we talked about survivorship curves. Do you remember that part? Better help you out here. What do I want to say here? Distribution. And we talked about survivorship. Do you remember survivorship curve? Do you remember the different curves? 1 through 3. Oh, look, that's a really cool way of writing it. 2 different ways. Miss Litton needs some toast. Okay, 1 through 3. So what, do you remember when we looked at those? That you always start out at like 1,000 individuals. Remember this? Okay, and what survivorship curve are we?
1.
We're type 1, okay, which means most of us die when we are what? Old. And then type 2 was like
songbirds.
Yeah, and any time. And then type 3, what happens with them?
Die out quickly.
Yeah, most die young. These are like a lot of plants. Most of them don't survive. So we talked about survivorship curves, and then we talked about regulation and types of growth curves. Do you remember the 2 types of growth curves that we discussed? Yes. What are you going to tell me? J-shaped growth curve. So this is time, and this is number of species for both of these. And then what was your other one? S. Okay, and what's right here?
Carrying capacity.
Good, the carrying capacity. And we said both of them have exponential growth, but then they try to level off. We also talked about density dependent. Things like competition, amount of and food and mates and habitat where you live. these types of density dependent factors usually are what regulates those S-shaped growth curves. Okay, where there were density independent, what are some density independent factors?
Weather, natural
Yep.
disasters.
Disasters, weather, okay, those types of things usually regulate a J-shaped growth curve. All right, and we talked about human population, and I'm gonna add these on here for you. On human population, what did we talk about for that?
LDCs, MDCs.
Perfect. See, you remember it all. Okay, more developed and less developed countries. Remember that, and how they show up. We looked at versus. Remember this?
Yep.
So which one is a more developed country? MDC.
Column.
Yeah, the column. Okay, and we talked about these are pre-reproductive, these are reproductive, and these are post, right? And if you have a large amount of people like in an LDC that are entering into the reproductive, then that's gonna be a lot of people increasing the amount of babies you have, and what's gonna happen to
Growing
that
population.
growth? Yeah, they are definitely gonna grow, where this population is gonna stay more stable. Okay, also in chapter 33, we talked about the types in the community, things like organisms in the community. You have producers and what else?
And consumers.
Consumers, what else do you, okay, good. So let's break down the consumers. Do you remember the different types of consumers that we had?
Primary, secondary.
Primary, secondary, and tertiary, good. So make sure you review the whole producers, consumers, and decomposers. How many of you have hit that in your Octodoc already, this topic? Okay, so you'll get to that. We talked about competition, we talked about predation, we probably talked about, did we talk about symbiosis? Yes. So we talked about that some organisms, you have the predator-prey relationship that cycles. Predator-prey. So as the prey numbers go up, usually the predator number goes up, right? And then as the predator number goes up, then what happens? Yeah. The prey number goes down, and then so too will the predator number. We talked about that cycling. We talked about how to be a harder prey. We talked about ways to hide, right? You have something for me? Maybe you're gonna look like a rock or something like that, right? We talked about those issues. Then we talked about symbiotic relationships. Thank you, sir. And everything's submitted online?
Yep.
Okay. Do you remember the 3 types of symbiotic relationships? Yeah.
Commensalism,
Commens.
mutualism, and I can't remember.
Starts with a P. Parasitism. Remember, it's always good for one of them, right? In that relationship, somebody it's working out for. In parasitism, it's good for one and it's bad for the other. In mutualism, it's good for both. And in commensalism, what is that about?
Oh, isn't it like one benefits and the other one isn't harmed?
Yep, one benefits and the other one isn't harmed. That's good. And then I think, I don't have my notes right in front of me, but we talked about, did we talk about keystone predators at the top and how, does that sound familiar? I don't want to overspeak our class. Go ahead, what were you going to say?
Is that like how being at the top isn't good all of the time?
Oh, we did talk about that. Yeah, we probably talked about how when you're at your, that was more in pecking orders and behavior, how you put yourself at risk. But we talked about, here, I'll look right here. No, we didn't talk about that. Okay, so we talked about that a little bit of disturbance is always good in a community, kind of shakes things up a little bit, like something, what could something happen like in a forest? Yeah, yeah, fire. that can be good because it can kind of shake things up a little bit within that community.
You're recycling?
If I do, it's in the back of the classroom. That can be good. We talked about primary and secondary succession. Primary succession is when you're starting on plain what? Rock. And secondary succession, remember secondary succession was soil? Okay, good. Okay, so those are some key things in chapter 33. Okay, and if you need to, you might review a few of those things. Okay, and then the next chapter we talked about was chapter 34. Okay, and in chapter 34 we were talking about the nature of ecosystems. Oh, and you know what? I jumped. Did I jump a little bit? Oh, this is your autotroph, heterotroph. Sorry, I just went right into it here. So let me put that here. Producers, consumers, that was in this chapter. Sorry about that. Are you looking for it and going what? Here we go. Paste. Okay, so that was here in chapter 34. See, we already learned one thing. We're good. We're ahead of the game. All right. Then we talked about energy flow and chemical cycling. Energy flow and chemical cycling. Does anybody remember what that is about when I say that? Energy flow and chemicals. Yes.
How it's not 100% efficient because as you go through the energy levels, that you're gonna lose 10% of your energy, and that's why the bigger animals need more.
Right. Exactly. So we have fewer and fewer as we move up through the food chain because not everything transfers over, and that sounds. Yes, that's good. And then when we talked about energy flow, where's the energy all starting from?
The sun.
The sun, and then it's moving through the organisms, exactly, and about 10% passes each time. And then chemical cycling, if you recall, we reviewed a few cycles, right? And we'll go over those as well. We talked about the different cycles, the phosphorus cycle, nitrogen cycle, carbon cycle, water cycle. But before I move into those cycles, do you remember the difference between food chains and food webs?
Chains is one direction, and the webs can go to kind of many, I guess.
Yeah, food chains follows one piece of food all the way through, just directly through one pathway, where food webs they're interlinked. Many different food chains in a food web. Okay, we also talked about trophic levels. Remember, if you're eating at the first trophic level, do you remember what you're eating at? What's the lowest level you can eat at? A salad, right?
Yeah.
So that's at the first trophic level, or autotrophs, right? And if you're eating at the second trophic level, that would be like cow, because you're eating a primary consumer, right? And if you eat something that eats a cow, if you eat fox or wolf or something like that, then you're eating at the third trophic level. So I think of it like what's in my trough. So if I'm eating at the lowest trough level, it would be plants. If I eat at the next trophic level, then I'm eating something that eats plants. If I'm eating at the next one, I'm eating an animal who eats animals who eats plants. And that's where we could stack those pyramids. Remember, the pyramids got smaller and smaller, which is what you were referring to, okay? So those were your ecological pyramids. And then we have our biogeochemical cycles. And we talked about the water cycle. Guys, not too loud over there, okay? Guys, hi, hello. Not too loud, okay? So we talked about the water cycles, and in each of these cycles, we talked about what they are, how they move through the cycle, and then how man can screw them up. Pretty much you can put pollution on every one of these cycles, and you would have one thing that you could say how man screws that up, works every time. On the water cycle, we talked about words like evaporation, transpiration, evapotranspiration. Does that sound familiar from your last year too, right? When it evaporates out of plants. Precipitation, those are all those types of things. Then we talked about the phosphorus cycle. And do you remember where the phosphorus cycle, where the reservoir is for that?
The earth,
Yep, good, in rocks. And how do we get it out of the rocks?
runoff.
Yes, the word you want to use is weathering. Yeah, so wears away the rocks, good. And then usually phosphorus is a limiting nutrient, And so as you, as it gets generated, it gets immediately brought into living organisms, and so it's usually a limiting factor to inhibit growth. And we talked about one of the ways man can really screw things up is by putting phosphates from soaps and runoffs into water, bodies of water, and it can cause over-enrichment of a lake, like eutrophication, etc. Okay, we then talked about the nitrogen cycle, and we have a whole little song online how we get the nitrogen right out of the air and into a solid, nitrogen fixation, where you take it from the nitrogen gas N2 into ammonia, then nitrification. First you form what? Nitrite, then you form nitrate, right? And then from nitrate you serve it up to plants. You can re-put it up into the atmosphere, into the reservoir through denitrifying bacteria, or if it's brought into a living organism, you have decomposition, right? And then after that decomposition, it goes back into ammonium again. Okay, and last but not least, we talked about the carbon cycle, and then basically there you're seeing the connection between photosynthesis and cellular respiration, but what kind of things also generate CO2 into the air? Man through combustion, yeah, burning of fossil fuels, and also in two ways, because we burn fossil fuels and put additional CO2 in, and then we cut down plants, which, you know, takes away some of our abilities to remove CO2 from the air. Okay, so we hit it on multiple levels. Okay. And then the next chapter we talked about in Unit 1 was Chapter 32. Okay, and Chapter 32, if you recall, that was all about behavior. And so the first things we talked about, let me just get something here so I'm the same font. Okay, so we talked about nature versus nurture, and if behavior, remember we talked about it, whether it was in our genes or if it was due to our environment. That was the whole nature versus nurture. And you remember we had experiments for both of those. We talked about, if we said it's in our nature, in our DNA, we talked about like lovebirds and forming those hybrids, remember? And they would have long and short pieces of nest material, right? And then if the hybrid had an intermediate and would fall out of its tail feathers, we looked at those snakes and preference for slugs. And we had a hybrid who had an intermediate affinity for slugs, and we looked at twin studies and saying that behavior could be in our DNA. Then we looked at the environment influencing them. We talked about fixed action patterns. We talked about birds learning the song of their parents, if you remembered. And all of that was underneath imprinting, that usually at a specific time in your life or life cycle that you can learn new materials. And then we talked about associative learning. Write these words down. Okay, so imprinting, here, I'll put it in here, there's a sensitive period. When you can learn, sensitive period, when you can learn new information. And then we talked about associative learning, where, what do you hear in the word associative? What? Associate, so you're associating two things, good. So we talked about classical conditioning and operant conditioning. Remember classical conditioning, our example there was Pavlov's dogs, and operant conditioning was Skinner's rats, where you reward them following, girls? You reward them following a behavior, whereas like it's about rewards and punishment, where when you're doing classical conditioning, it's a physiological response, like salivating according to food. And then we talked about mating behavior. So we talked about female choice, like why would she choose? And we had some different hypotheses there. Was it based on good genes, like they could provide a lot for their offspring, or were they good looking, which was the runaway hypothesis, good genes? Or we talked about how males and females can look different from each other. We talked about territory. We talked about sexual dimorphism, where the males and females look different. And we talked about sociobiology and things like altruism. Do you remember what altruism is?
Isn't it where you like sacrifice yourself?
Yes, like, are you going to sacrifice your genes for others? And a lot of times that was, people thought an organism was being altruistic, but they were really protecting their what? Offspring. Because your fitness is based on your ability to pass your genes on. And so if you could increase the odds that your genes would get passed on, it wasn't really a sacrifice that you were doing. We talked about, did I talk about a reciprocal altruism, like you scratch my back, I'll scratch yours? And we looked at vampire bats and how they would give a blood meal to their neighbor if they didn't have it, but if they were a cheater, then they would stop feeding them. And then we talked about communication. Do you remember the different types of communication? Yes.
Auditory, visual,
Auditory.
tactile.
Good. Visual, tactile, another one.
Like sight.
Chemical. Yeah. Visual would be sight.
Oh.
Okay, and being familiar with some examples of that. Okay, that was Unit 1. Okay, moving on now into Unit 2. In Unit 2, we had Chapter 1. This up here. Okay, and in Chapter 1, we talked about life. And we talked about the characteristics of life. Living things are what? Yeah, made of cells. Good. So you remember all that. And we talked about the organization. What is organization about? Atom, molecule, organelle, cell, tissue, right? Remember that whole bit? Okay. We talked about classification categories. I don't know what I'm spelling. Do you remember kingdom, phylum, class, order, domain, kingdom, phylum, class, order, family, genus, species? Okay, classification. We talked about domains. How many domains were there?
3.
3. Okay. What were the domains? Bacteria.
Archaea.
And eukarya. Good. And you just want to differentiate between those as well. I introduced the organization of the biosphere, which we already had learned it. This is Chapter 1, but you had already done all of the ecology about biodiversity and chemical cycling versus energy flow. But we did bring up the biosphere, and the biosphere is where all life exists, is in the biosphere. And we talked about the process of science, and that would be like your scientific method. So we talked about asking questions and developing models. We talked about those and using math. Those would be the science practices. But we also talked about the scientific method. Make your observation, write a what? Hypothesis, right. So you want to review that as well. And then we talked about technology and the application of scientific knowledge. Does that seem like forever ago?
Yeah.
No, right? Okay, and then Chapter 2. Chapter 2 was basic chemistry. And so in this one, things that you want to review are atoms. So do you remember the atom story? Atoms are made of what? Protons, neutrons, and electrons. The protons and neutrons, right? So you want to know things also like organization. Okay, we talked about protons, neutrons, and electrons and where they are. We talked about bonding. What are the different kinds of bonds we talked about? Covalent and hydrogen bonds.
Thank you.
You're welcome. And in covalent bond, remember we had polar and what?
Non-polar.
Non-polar covalent bonds, good. And then the hydrogen bonds, that was really important when it came to water, right? So we talked about just properties of water. Okay, that it has a high boiling point. What are some other properties we talked about?
Adhesion, cohesion.
Okay, good. Then we spent a bunch of time talking about organic molecules. Organic molecules, we talked about, do you remember what they are? Of course you do.
DNA.
Carbs, proteins, right? Lipids, and nucleic acids. And we talked about the monomers, and then those were the polymers. Remember, we talked about ways to join them. Remember dehydration?
Synthesis.
Yep, synthesis.
Monomers form polymers.
Yes, monomers form polymers. Right? That makes sense, right?
Yeah.
Dehydration, synthesis to join versus what was the other reaction?
Hydrolysis.
Yep, good job.
I have a quick question.
Yes, go right ahead.
For monomers, would the monomers be nucleic acids forming DNA kind of?
The monomers would be a nucleotide
Okay.
forming a nucleic acid.
Okay, so like-
So these right here, these organic molecules, carbs, proteins, lipids, and nucleic acids, these are the polymers. Right?
oh, yeah.
Ah. The polymers are that. The monomers are gonna be things like, are gonna be sugars, right? Fatty acids, glycerol, a nucleotide, an amino acid, right? Those are all the monomers that join to form the polymers. Okay, and then that brought us to chapter 3. Okay, and let me just move this up here a little bit. Okay, and then chapter 3. Okay, chapter 3 was about the cell. So we had the cell theory, that all living things are made out of cells, that it's the building block of all living things, and cells only come from other cells. And then we talked about prokaryotic versus eukaryotic cells. We talked about organelles that are found in eukaryotic cells, right? And we went through the structure and function of all of those organelles, so you're probably going to want to review that just a little bit. That's like a straight up memorization aspect to it. Yes, I'm listening to you.
Organelles, do you mean like the like thing, not things, but the structures inside the cell? So like the nucleus
Like
or something like that?
the things I would think there is like that the lysosome,
Okay.
you know, equals the stomach of the cell.
Okay.
So I'll do a
That's
for
good.
example there.
Thank you.
I'm not doing a for example. Lysosome equals stomach. Okay, and so on that, we talked about the nucleus, we talked about the endomembrane system, vacuoles, the energy-related organelles, and cytoskeleton. Okay, so that one you just want to go through and review each of those. Maybe look at a picture. You probably have you could Google, you know, a picture where all the organelles are identified and just review it that way. I think that'd be the easiest thing to do. I'll put Google a pic with organelle functions identified. That would be a really good, easy way to review that chapter. Okay, and then our last chapter of units 1 and 2, chapter 4. And chapter 4 was all about the cell membrane, if you recall. So for this one, we talked about, membrane structure. Remember things like your phospholipid bilayer and the proteins, right? So membrane structure, we focused on protein functions. You remember these. Channel, what? Carrier. Cell recognition, receptor, right? Enzymatic, good. Junction, so you want to review those. So functions of proteins. Then we talked about permeability of the membrane. Right? We said, what are the 3 ways to cross? Through the phospholipid. But you couldn't be what? Large or charged. What else? Channels or carriers, and what? Whole membrane. Now, when you talk about through the phospholipid, this is when you're talking about osmosis, when you do through the phospholipid. And so we talked about osmosis. We talked about hypertonic. Remember these words? Hypotonic and isotonic. So things like when you look at a solution, whichever has, let's say, the most sugars in it, that, you know, compared to another solution. Remember we said it's always relative to another. If this is up, then this is down. So if this solution on this side of the membrane is hypertonic, the other solution must be hypotonic. So a hypertonic solution has, like, let's say, a bunch of sugars in it. Then the neighboring solution by default is hypo. And when you talk about osmosis, that's the diffusion of water across a semi-permeable membrane. We always said water must flow from the hypo into the hyper. So if I take carrots and put them in distilled water, do you think my carrots are going to get bigger or smaller? Bigger, because the water will flow into the carrot, right? But if I put carrots in salt water, the carrots will probably what? Shrivel up. We talked about how pickles used to be what? Cucumbers put in salty solution. So you're just following the water, okay? Following the water, and which way would it go? Okay, and then we also discussed passive versus active transport. So passive transport does not require any what? Passive does not require any energy, but active does. So when would you use active transport? When you go against the concentration gradient, right? Or whole membrane. And whole membrane would be what kind?
Endocytosis?
Yep. There was exocytosis and endocytosis. And of endocytosis, there was pino and phago and receptor mediated. Remember that. Okay, and then that was it for unit 2. So my strong suggestion is you have a limited number. I mean, you have the honors final starts with period 2, so that starts a week from tomorrow, right? A week from tomorrow. So your idea, what I would do is, of these, we just reviewed 7 different chapters just now, right? 7 different chapters we just reviewed. Of the ones, you've taken quizzes over a lot of those. So of the ones that you had the best quiz grades on, just cursory, reviewing. Focus on the ones that you had the lowest scores on. And the best ways to review is obviously your Go papers, and they're going to have all the details. But even taking your book and just flipping through all the pictures in that chapter, you'll go, oh yeah, I remember. Or just looking at all your pictures that you flipped into your group shared notes, those are going to help you remember things. And then you know that the written part of your final is your Octodoc, right? So that's half of your final. That's 100 points of your final is your Octodoc. The second half is going to be multiple choice. So preparing, the Octodoc you're doing in class, you have lots of feedback on every step. I'm expecting you to get 100 because people are always evaluating your work on it, right? Every time you do something, somebody's going to do an SOS on your slide and you're going to quickly correct it, right? But the multiple choice part, the best way to prepare for a multiple choice is to take what? Multiple choice tests. So do things like maybe go in the back of your book to the multiple choice questions at the end of the chapter and just do all the odds for that chapter. If you get a really good grade, call it good. If you don't have a good grade, if it's not a grade you want to see on your final, go back and read through the group shared notes again, right? Because we've just kind of hit everything briefly on these 7 chapters. So go back and read through the notes again and now take all the evens. Now you have another set of questions you could ask yourself multiple choice wise, right? And if it's a question that they ask, but I don't have it in the group shared notes, should you stress out about it? No, if it's not in the group shared notes, if it's not one of our terms, don't freak out about it. Okay, so you can always use that as a guiding principle. If it's one question, you're like, I don't even remember learning about that. Well, then we probably didn't learn about it, right? It's not something I had you focus on. Okay, so you have 7 of them. Go through, do the multiple choice at the end. Maybe you look through all your pictures first, but I think you just got I'm proud of you because you sat, think about it. We spent more than 2 months just doing those 7 chapters and we just did them in, 36 minutes. All right, so go back and review. Super proud. Any other questions? Yes.
Can you post that?
Yeah, sure. I will take this make it a PDF and put it with the YouTube link.
All right.
Okay?
Thanks.
All right, no problem. All right, make good choices, you guys.
Thank you.
Do you want me to Instagram these?
Yeah.
Okay, you got it. If you're up late, have a piece of toast.